Hello everyone, thank you for joining me today in this talk that will be about the in vitro characterization of serotonin 2A receptor agonists by NanoBiT Assays. First of all, thank you for having me here in Berlin, and thank you for letting me speak about my research. Okay. So what does this presentation consist of? First of all, I will say something about why we were interested in developing such an assay, how we have set it up. Then I will briefly go over two applications, or not actually very briefly, but first application on structure-activity relationships, and secondly on psychedelic or comparison between psychedelic and non-psychedelic ergolines. Then just a brief introduction on some more applications that we're working on to finish off with a broader perspective and an outlook on those assays. So first of all, why were we interested in having such assays that I'm coming to talk about? So I may have not introduced it on the introduction slide, but I actually come from a laboratory of toxicology where a recent challenge throughout the past few decades in toxicology has been the emergence of new psychoactive substances, which are substances that are not the same as classical illicit or whatever psychoactive drugs, but they have the same effects. They are designed to mimic these effects, but they have different structures. So if you like to read the definition that is given by the United Nations Office on Drug and Crime is on the slide, but it's obviously a legal definition. So if you look at the first pointer, if you look at the distribution of those new psychoactive substances, you can see that there's different colors. So there's different groups of compounds depending on which effect they have. For example, synthetic cannabinoid receptor agonists, they are compounds which are supposed to mimic the effects of cannabis. And classic hallucinogens, which is a bit of a counterintuitive name for new psychoactive substances, are designed to mimic the effects of LSD, mescaline, psilocybin, and so on. So despite what you may think, if you see classic hallucinogens, these are also a large group of substances which actually all have the same mechanism of action, being activation of the serotonin 2A receptor. So this is the group in orange, and you can see that there's actually consistently throughout the years quite a lot of compounds which are present on the illicit drug markets or not so illicit drug markets. So these constantly changing structures, they pose issues in terms of their detection, because typically in a toxicological lab, you look at specific structures in which you're interested. But most importantly, and also what we are going to talk about today, is the fact that their effects are actually unknown. So if one of those modified structures reaches the market, it hasn't been tested anywhere near where a typical drug is tested. It may be that this compound is entirely inactive, so then there's no issue actually. It may be that the compound is has about the same activity of the compound that it's mimicking, which is also still relatively okay, but it may be that it's much more potent or efficacious, which makes that its effects are maybe potentially dangerous. So if I say something about these different structures, what am I talking about? So there's three structural groups actually of psychedelics. Typically are phenylalkylamines, which are structurally similar to mescaline. So this is the basic structure of mescaline. Then there's tryptamines, which are structurally similar to psilocybin and psilocin. And then there's the ergolines, which are actually conformationally restricted forms of the tryptamines, of which LSD is the prototypical example. So for tryptamines and phenylkylamines, there's quite a lot of structural variety described in the books by Alexander Shulgin, for example, Tikal and Pikal. For ergolines, it's more difficult to change the chemical structure and still obtain a compound which is active as a psychedelic. So they have very different structures, but what they have in common is the fact that they share serotonin 2A receptor as a pharmacological target. There may be others. For example, for tryptamines, it may be the serotonin 1A receptor. For the phenylalkylamines, it may be the serotonin 2C receptor, and so on. So we started from that serotonin 2A receptor, and the purpose of the assay that we aim to set up was to estimate the presence of psychedelic effects in newly detected substances. So if we get a compound which is newly detected, can it have psychedelic effects, yes or no? And what we were also interested in, for the ones that we already knew, was to characterize their structure-activity relationships. So for that, we set out a few requirements of that assay. It's supposed to be specific for the serotonin 2A receptor as a common target of those psychedelics. And for us, just because it's easiest like that, we wanted it to be sensitive, easy to use, and useful in a relatively high throughput manner. So how did we go about this? So if you look at the serotonin 2A receptor, you can see that it has 7 transmembrane domains. It's a G protein coupled receptor, which is a type of receptor which translates a signal from outside the cell to an effect at the inside of the cell. And actually quite a few intracellular effects or intracellular interacting proteins have been identified throughout the years, where mostly people look at the activation of the G-alpha-Q pathway, which then induces IP3 activation and calcium or generation and calcium release. Throughout the years, also people have looked at arachidonic acid release and at beta-arrestin 2 recruitment to the receptor. So there's obviously quite a few other signaling pathways which have been linked to psychedelic activity or not psychedelic activity, just activation of the serotonin 2A receptor. But those are more difficult to quantify and are not typically looked at. So the ones in the blue frames are the ones which are signaling effects that are typically monitored for psychedelics. So what we were considering when we chose an event to monitor for the serotonin 2A receptor was, first of all, the fact that when you start from an event which is upstream signaling cascade, which means that it's still relatively close to receptor activation, not in terms of the localization in the cell, but just how fast it happens after activation of the receptor, is that when you go further downstream, no, I'm lost in the middle of my sentence, I'm sorry. So if you stay upstream, you get little signal amplification. If you go further downstream, the signal gets amplified and amplified and amplified, so you get a stronger signal. This is interesting on the one hand because it increases the sensitivity of your assay. On the other hand, it may also lead to the fact that it's difficult to distinguish a partial agonist from a full agonist because everything just reaches a very high effect. Then also something else that we were considering is the fact that we typically look at those in vitro assays like it's a cell and it expresses only one receptor. However, this is definitely not the case. So there are definitely other GPCRs present at the cell membrane. There's also other types of signaling or whatever other proteins in the membrane that are present which can interact with compounds that you are adding to the cells. Which may lead to, for example, also activation of those signaling pathways, which may lead to altered levels of serotonin and so on. And the further downstream you go, the higher the effect is that there may be a signal interference, for example, by other GPCRs. So both of these, the increased signal amplification and the increased signal interferences, actually led us to conclude that it would increase the complexity of the interpretation if we would look at an effect further downstream, even if it may be more sensitive. So therefore, we selected 2 upstream events, being the interaction of the G protein with the receptor and the interaction with beta-arrestin with the receptor. And the selection of these events actually ensures receptor specificity, in our case, as you will see later, it's only possible to monitor activation of the serotonin 2A receptor and not of other receptors. The selection of the technique, being the NanoBiT technique, ensures the sensitivity. What is the NanoBiT technique? NanoBiT stands for nanoluc or nanoluciferase binary technology. And this is actually a nanoluciferase, so a small luciferase enzyme, which is split into two parts, hence binary. And when those two parts, the small bits and the large bits, are separate from each other, they are not active, so the enzyme is not functional, so there can be no light that is generated. If the two parts are in close proximity, it can lead to functional complementation of the enzyme, which means that you can pick up or catch a light signal. And this light signal is actually very sensitive because unlike fluorescence, there's little or no background luminescence in cells. So how did we do this? So we fused one part of the nanoluciferase enzyme to the receptor, which is here for the one assay and here for the other. And then the other part was fused to either beta-arrestin molecule or to a mini-G protein, which is an engineered version of the G-alpha subunit. We used the engineered version, which was, by the way, not engineered by us, because it has a more stable interaction with the receptor, because the interaction between the G protein and the receptor is usually very short, so it's very difficult to capture that. So when no agonist is present, those two are, in theory, separated, and there should be no luminescence. If then an agonist, it can be psychedelic or a non-psychedelic agonist of the receptor, interacts with the receptor, the intracellular protein gets recruited, which means that it's functionally restored and that you can measure a luminescent signal. So the assay protocol is a 4-day protocol, where on the first day the cells are seeded, these are hex cells, so an immortalized cell line, are seeded in 6 well plates. They are transfected with the plasmids which encode for those fusion proteins. Then they are reseeded on the 3rd day in a 96 well plate, and then there's the readout, the cells are washed, there's a substrate for the enzyme that is added, and the agonist is added, which is then upon which the plate is transferred to the machine, which is called the TriStar, but it's just a luminometer, and then we read this out for 2 hours. So what do we get from that is concentration response curves, which have a sigmoidal shape and from which you can derive certain pharmacological parameters. So first of all, from the height of the signal or the height of the curve, you can say something about the maximal effect of the compound that you were measuring, or the efficacy or the Emax, which actually corresponds or says something about the strength of the effect that such a compound can induce. For example, in this case, the black curve is more efficacious than the gray curve. A second one that you can derive from this is the EC50 or the potency, which is actually the concentration of a compound that you need to get half of the maximal effect of that compound. So, for example, here, the black curve will need a concentration of something like 10 to the minus 8 molar to get half of its maximal effect, whereas the gray one will need something like 10 to the minus 6 molar, so a much higher concentration, which means that it's less potent. So now, of course, it's quite a step to go from the luminometer to the concentration response curves. So just to be clear on how we got there, so we ran these compounds in the assay. If you get an inactive compound or a solvent control, you get a slightly decreasing signal, like the one in gray. If you get an active compound, you get an increasing signal, after which it also goes down for a bit. So we then calculate the area under the curve of that pink curve and then subtract the background control. And we always say percentage AUC because we express this relative to a reference compound, which for the purpose of this presentation is always LSD. We've also compared it to serotonin, but this would lead me a bit too far if we discuss this today as well. So first application that we used this assay system for was to assess structure-activity relationships. So the aims that we set out was first to gain more insights into the in vitro activity of the molecules. We wanted to know whether certain structural features could be linked to changes in activity. And furthermore, we were also interested to estimate whether or not the in vitro data that we obtained were correlated with doses that are typically reported by users of those substances. So if we look at the structures of psychedelic new psychoactive substances, we looked at the phenylalkylamines. More specifically, one of those compounds is, for example, 2CB. If you look on the internet, you can, for example, find such pictures. So it's supposed to be psychedelic. It's also a bit entactogenic, so it's not entirely psychedelic, but it can be categorized as a psychedelic. If you add a methyl group to this, you can get, you get compound DOB, which is actually an amphetamine. The basic structure here is called an amphetamine. For this, you could find on the internet user reports which state my DOB nightmare, so that may be a bit stronger than the other one. And then if you add an entirely bigger group still to that structure, you get 25B NBOMI, and if you look that up, you get a warning that it may be fatal at heavy doses. So just to know what we were looking for or what we were expecting. If we have a look at those structure-activity relationships in our assay, so we have the response in the y-axis, the concentration in the x-axis, and we always compare to the reference LSD, which is shown here in gray, has an Emax or a relative Emax of 100%. If we add to that curve the structure of, or the curve of 2CB, we can see that in orange, the curve is a slight bit lower and a slight bit shifted to the right, meaning that you need a slightly higher concentration and you get a slightly lower effect in vitro. But actually, I don't think that this is a statistically significant difference, so it's more or less the same. If you would then have a look at DOB in blue, you can see that actually it's also relatively similar to LSD, but the curve is a bit higher and a bit more shifted to the left, hence you need a lower concentration and get a higher efficacy. Definitely as compared to 2CB. Then for 25B NBOMI, if you add that curve, it's quite a bit of a shift because it's much higher, so it's much more efficacious than all three other substances, and it's also clearly shifted to the left, so you need the lowest concentration in vitro to get half of the maximal effect. We didn't limit ourselves to these three substances, so we didn't only look at a compound which had a bromine at position X. We also had a look at compounds with just a hydrogen atom, a chlorine, an iodine, a methyl group or an ethyl group, which are all actually psychedelics, except for the one with the H, with the hydrogen. So again, we had a look at the comparison in those concentration response curves. We had a look relative to the reference, which is again shown in gray everywhere, is just a curve of LSD for comparison. If we add in orange the curves of the 2CX compound, we can see that we actually saw the same as for 2CB. In most cases, it's shifted just a bit to the right, so you need a slightly higher concentration, and it's shifted just a bit below LSD. With the exception of 2CH, which is remarkably shifted to the right and also remarkably lower. We had the same for the DOX substances, so we add in blue dose curves, which again follow the same pattern as before, so they're a bit more potent, a bit more shifted to the left, and also a bit more efficacious, so a bit higher curves. And then there's the NBOMIs, which are then again shifted much to the left, so much more potent, a much lower concentration is needed, and a much higher effect is reached. So obviously this is all very interesting, but we were also interested to know whether or not this in vitro activity was correlated by doses that are reported by users. And for this, we had a look at a few websites, such as Stripsit, which for each of those substances, or maybe not each of those, but quite a few of them, then states, which if you do oral intake of this compound, what is a common dose or a light dose or strong dose or whatever. So we always had a look at the common dose, and since different sources give different common doses, we also had a look at other sources. For the 2CX and the DOX compounds, there was quite some information in PICAL, for example, and then here a layout which may be quite familiar. So this is Psychonaut Wiki, which uses the exact same layout as Wikipedia. Also here there's common doses. So we then continue to plot the dose that we found relative to the concentration that is required to get half of the maximal effect in the assay, so the EC50 value. If we did that for all these compounds, you get quite a few compounds, and actually you can already see visually that you could draw a line between them. And also, when we did the statistics on this, we could see that there was a clear correlation, so that was nice. And also interesting to know is actually the fact that those compounds, or the groups of compounds that we had previously seen, that they were also recognized in this correlation. So here, the 2CX compounds, which has had a relatively high EC50 value, they had also the highest dose at which they are typically used. The DOX compounds with the extramethyl group, they shifted a bit to the left, so a slightly lower dose is required, and also they had a slightly lower EC50 value. And then the NBOMIs are used at much lower doses and also had much lower EC50 values. So there's a few compounds which are floating outside these groups. For example, here there's mescaline, which is not technically a 2CX substance because it has a methoxy group here instead of here, and then here one and here one. So it's slightly differently substituted. This had a very high concentration that was required to get activation in the assay, but this is also typically used at a very high dose. And then here there was LSD, which had a moderate potency in our assay, but is typically used at a very low dose. This may be because this is obviously a different functional group, or different structure actually, so they may have a slightly different correlation. So those were the aims that I started with. We can now get to the conclusions of this part. First of all, the beta-arrestin recruitment assay could be used to generate in vitro concentration response curves, where the general pattern of both potency and efficacy is that 2CX substances were less active than DOX substances, which were both much less active than 25X NBOMIs. And we could also say that the in vitro activity was correlated with the reported dose estimates. So a second application that we then had a look at, if we saw that, was we had a comparison between psychedelic and non-psychedelic ergolines. So what did we want to look at was, first of all, we wanted to gain more insight into the in vitro activity of the molecules, whether structural features again could be linked to changes in the activity. So this is actually relatively similar to one of the aims that we set out for our previous application. And we definitely wanted to estimate whether or not different in vitro activation patterns could be obtained for psychedelic agonists of the serotonin 2A receptor and non-psychedelic ergoline agonists of the serotonin 2A receptor. So we're looking now at a middle group where a rather small structural change may result in the compound shifting from a psychedelic substance to a non-psychedelic or the other way around, but usually towards the non-psychedelic side. One example here that is typically used when comparing psychedelic with non-psychedelic substances is the comparison between lizarite and LSD. So lizarite here is an additional nitrogen atom, which is also a bit chemically oriented a bit differently versus LSD here, but actually these structures are very, very similar. Yet lizarite, which activates the serotonin 2A receptor, doesn't have psychedelic properties and actually is used as a drug in Parkinson's disease, whereas obviously LSD has psychedelic properties, also is an agonist of the serotonin 2A receptor and is subject of certain legislative measures. It doesn't stop at that, so there's other non-psychedelic ergolines, there's other psychedelic ergolines. So for the non-psychedelic ergolines, we had a look at 2-bromo-LSD, which is also called BOL sometimes. There's a few papers that have recently come out which are actually looking at potential antidepressant effect of these compounds without having psychedelic properties. Then there's on the psychedelic side ALLAT, which here has a another different chemical group than here, just the methyl. This substance has emerged on the drug market as a new psychoactive substance and has then been controlled. And then there's other substances for which actually it's not very clear what the effect is. For example, LAMPA here has a slightly different substitution pattern on that nitrogen atom. So actually some people report that it has some effects which remind of LSD, others say that it doesn't have any effect at all. So it's a bit inconclusive, but probably it won't be the strongest and most outspoken psychedelic if you doubt whether it has psychedelic properties. What did we not include in this study was prodrugs such as CP-LSD. Actually, a lot of ergolines which have been appearing on the drug market are substances which are just metabolized in vivo to LSD. So it means that it's not that compound in itself that activates the receptor, but LSD. So for the comparison, we now use the two assay formats, the mini-G assay, the beta-arrestin assay, and we did that the same way as we did it before. So on the left is the mini-G assay, there's the curve of LSD to which all else is compared, and then on the right is the curve for beta-arrestin with again the comparison to LSD. So first we had a look at ALLAT. Here there's some specifications, some technical details about how it's supposed to be used or whatever. If you add the concentration response curve of that to that of LSD, you see that it's actually more active than LSD at the receptor. It has a potency which is more or less similar to that of LSD, but it has a higher efficacy actually in both assays. Then there was lizarite, which was not psychedelic. If you add the curve of that compound to those of LSD, you can see that there is both a shift to the right, so it means that it's a bit less potent than LSD, also here, but definitely that there is a shift in the efficacy of the compounds, so the efficacy was definitely much lower than that of LSD. 2-bromo-LSD, so the potential antidepressant. So if we add the light blue curve to that of the other two, we can see that it behaves in terms of efficacy rather similar to lizaride, so very low in both assays or halfway LSD, but it's a bit more potent, so actually the potency is relatively similar to that of LSD. So now again adding a lot to the comparison. If we now have a look at LAMPA, so which may have little or no noticeable effects or little effects, yeah, who knows. If we add the curve of that to the comparison of the other compounds, we can see that it's right below the curve of LSD. So it actually has a very similar potency. Also, the efficacy is similar, but it is lower. So if we look back at the aims that we set out in the beginning, we can formulate the conclusions. So the reportedly psychedelic substances reached higher receptor activation than the reportedly non-psychedelic substances in both assays relative to LSD. The current in vitro experiments suggest the existence of an efficacy threshold. So a minimum level of receptor activation is required for a substance to induce psychedelic effects, because the psychedelic ones had a higher efficacy than the non-psychedelic ones. This is not a hypothesis that we first put forward. There has been another group which has said that a lot before us about DOM and Ariadne. So that's also in vitro, and now then, or similar or the same timing, I think, as we published it, then they actually set out that a certain number as the efficacy threshold in a certain assay. And then there were in vivo experiments in this set of experiments as well, where we used the head twitch response for serotonin 2A receptor activation, but also body temperature and the distance that the mice traveled, which are actually markers for serotonin 1A receptor activity. And there it was suggested that there was a strong serotonin 1A receptor agonism by lizaride, stronger than with LSD, which had an impact on the behavior of the mice. So then some other applications. So until this point, I only talked about what happens when you add one compound to the assay. Obviously, it's also possible to have several of them added. So something that we did was combine the effects of lizaride and LSD. So again, lizaride is here in purple-ish in both assays, and LSD was here in gray. So if we first pre-incubated the cells with lizaride and then added LSD, we could see that the effect went down. So at low concentrations of lizaride, LSD still reaches its maximal effect. So we added a fixed concentration of LSD, which was 10 to the minus 7, so it's right at the top of its curve. So when only a low concentration of lizaride is present, LSD can still reach its maximal effect. If you increase the concentration of lizaride, as you would expect, it brings down the effect of LSD. And this happens in both assays, where actually the maximal or the combined effect reaches then the final level of lizaride. Another application that I won't go into much detail on is biased agonism. So we obviously had more than one bioassay. So we had a beta-arrestin 2 recruitment assay, we had a mini-G assay. When an agonist interacts with the receptor, it may be that both of these assays are activated to the same extent. However, it's also possible that it has a certain preference towards one of those. For example, either beta-arrestin 2, which is activated to a much more outspoken extent, and mini-G to a lesser extent, or the other way around is possible as well. So what we did was we compared both of them in the two assay formats. And what you could, for example, do is combine the EC50 and the Emax values that you get in either assay into a bias factor beta, and then you get a plot like this. So this is just an example. The bias factor, because of the formula that you use, of LSD is 0, and then compounds which are more preferring towards beta-arrestin get positive values, and values or compounds which get negative values are more preferring towards mini-G assay relative to LSD. So that's an important note. It's always relative to LSD, since you use that as your reference agonist. If you use a different reference agonist, then you can only say something about the result relative to the other reference agonists. So it's not easily translatable. But yeah, we found some substances which were clearly preferring towards beta-arrestin 2 relative to LSD, being those 25H NBF, NBO, and NBOMI, for example. Which already brings me to my final part, the broader perspective and the outlook for this. So what we did here is we set up two bioassays via the same mechanism, but of course this assay has some advantages, but it also has some limitations. And actually at this point, each of the in vitro assays that is used has some advantages, but also limitations. So I prefer to be upfront about this rather than trying to highlight other people's assays' limitations and highlighting my own advantages. So what we see as the advantages of our bioassays is that we can use the same receptor construct for both assays. So the receptor is tagged with a protein, but it is tagged in the same way for both of the assays. We could use the same experimental setup and the same readout. So there's actually little variability introduced by different protocols that we use, and also there's no difference in the readout that you're using. So we're both times using luminescence, not once fluorescence and the other time luminescence or different fluorophores or whatever. So we only introduce small tags. The larger bit of the nanoluciferase is 18 kilodalton. For example, green fluorescent protein is 27 kilodalton, so that's much larger. The assay allows for a real-time readout, so it's not like you have to wait for 24 hours and then do one measurement and this is what you get. And the fact that we have an upstream event allows us to be certain that we are measuring serotonin 2A activity, because the serotonin 2A receptor is the only one that's tagged. So even if the mini-G or the beta-arrestin goes to another receptor, we're not picking that up. Disadvantages or limitations is the fact that we had to modify the receptor construct. So obviously there is something which is at the C-terminus of the receptor. This may impact its signaling properties, so it's not ideal. Even more modified is the mini-G protein, so it's engineered from a G protein, and then still there's a tag which is introduced. And also the fact that an upstream event taking place doesn't necessarily mean that it gets translated into downstream signaling. So actually, playing on those limitations, and specifically of the upstream event, which doesn't necessarily mean downstream signaling, we set out as a next step to validate these upstream signaling events by setting up new assays of signaling events which are further downstream, where we will use a combination of widely used assays and newly developed assays. So as a broader conclusion, we said that the current in vitro assays have a predictive value for the in vivo effects to a certain extent. All in vitro assays have inherent advantages and limitations. The perfect in vitro assay does not exist, or does not exist yet. The obtained results are definitely true in their specific systems, but the translatability to other systems may not be guaranteed. So it's always important if you report results from an in vitro assay that you specifically state which cells were used, which assay was used, and so on, not just EC50 and that's it. We have to be careful not to overinterpret the in vitro findings. So factors other than receptor activation are of major importance, obviously, to in vivo or clinical effects. So it's not, actually, it's nice that we saw a correlation between the EC50s that we got and those doses that were reported, but it would also not have been surprising if there would have been no effect at all, because obviously there's still blood-brain barrier penetration, there's metabolism, and so many other things. So when it comes to in vitro assays, there's more than meets the eye. And this is already my last slide, which took me 20 minutes shorter than when I practiced it at home. So thank you.